Hello, welcome to NEET World. Good morning to offline and online students. Myself, Laxman, I am from Vizag and I qualified CSR NET All India rank 172 and also qualified SET and NET examinations also. Most of you know me, but I am introducing myself because so much of students online is there. So, I am here to teach you the zoology topics for your EAPCET and NEET examinations, right? So, this platform you can learn the topics, understand the concepts, and you can clear the doubts whatever it may be in coming to the subjects for your examinations. So, utilize this platform and make sure yourself consistent to these classes and achieve your examinations. All the best, right? So, let us start today's class. Today I came with an important topic, that is animal kingdom. So, before going to the class, I want to tell you that this class, this animal kingdom is one of the most important topic where you get statement related questions and also direct questions, assertion reasoning kind of questions, and sometimes simply you will get matching questions also. So, this is the one of the important topic where you get scoring, where you have more number of questions, like for example you will get minimum of 2 and maximum of 3 questions you will get from this animal kingdom, right? So many people think why only 2-3 questions. So, as this is the core topic, you have to focus on each and every question, because each and every question counts and each and every mark counts. So, this is the one of the scoring topic. So, focus on this animal kingdom. It's a very simple topic and you have to remember many number of examples. You have to remember many number of characteristic features. So, I will teach you many number of examples also, and I will teach you how to remember characters of different animals such as organisms, like all the things, right? So, let us start today's class, animal kingdom. So, this animal kingdom we are going to study in 3 different classes. The first part is about basics of classification. The first part is basics of classification, and the second part is about non-coordinates, and the third part is about coordinates. We are going to study this animal kingdom in 3 different parts. The first part is basics of classification, non-coordinates and coordinates. So, why we have to study basics of classification? Because in non-coordinates, coordinates, there are so many terminologies are there. So, if you want to know about the terminologies, first of all you have to know the basics of classification. If you understand the basis of classification, then you will easily understand non-coordinates and coordinates. Am I audible to everyone, right? Right. So, let us start first basics of classification. As I said, we have to know about the basics of classification. So, first of all we have to know why we have to study the classification, why it is very important, right? So, in this world there are many millions of species are going to describe for every year, every time, right? There are many number of species that are going to present discovered in this world. So, all these species we have to identify the species, right? We have to identify, we have to classify the species, we have to place the species in a particular order. So, for that thing we have to study the classification. See, here I mentioned, so this classification is helpful in identification of species. When we discovered a new species, what is it? Is it plant or animal or what it may be? And we have to name the species. Yesterday you already studied, I think, ICZ and ICBN. So, these are the nomenclatures. Based on their rules and regulations, they will give perfect names to each and every organism. So, for that also we need to study classification, and also we have to assign a systematic position to a particular organism. Like for example, we have 7 different kinds of categories, like for starting with the kingdom. Next one is phylum or division, division for plants, phylum for animal kingdom. Next one, class, order, family, genus, species. So, these are the systematic positions where we apply to particular animal to place it in perfect position, right? So, further thing also we need to study classification. So the next question is, sir, what basis we classified the animals? On what basis? What is the method we used to classify the animals into different levels? So I want to tell that even though there are a lot of organisms which are having different structures, different functions, but we all share common characters. Very important, common characters. So based on that common characters only, we classified the animals into different levels, right? You see, the common characters are called as closely resemble characters. In each and every animal having closely resemble characters, based on that characters only we classified the animals into different levels. So let's see what are the characters. So those characters are called as fundamental features. So every multicellular organisms in this world shares common fundamental features. So you see, these are the fundamental features. The first fundamental feature is level of organization, body symmetry, number of germ layers, nature of coelom, body segmentation, and presence of notochord. So these are the features, fundamental features. With this, we classified organisms into different levels, right? So we have to study each level, all these things, and what are the classifications that are going to present in each and level, and we have to study all the definitions. We have to remember all the examples regarding in this classification. Okay, understand how many types of fundamental features? 6 types. Everything we have to study, right? You are writing, right? No problem. So this PPT is here only. You can watch later also. The first fundamental feature is level of organization. So this level of organization is about how an animal is made up of, how an animal is formed, with what the animal is formed. So in this level of organization, there are 4 kinds of levels, starting with cell to organ system. For example, the animal is made up of only cells, right? The animal is made up of only cells, then that level of organism is called as cellular level. See, you can see the cell loosely aggregates. Aggregates nothing but the clump together. The cells only formed to a particular animal. So that kind of organization is called as cellular level. So the best example in the cellular level is porifera sponges. So if you see the sponges is only unicellular organism. There is no tissue, there is no organ, there is nothing is there. Only cell is only present in porifera, right? So another statement I mentioned, you can see some shows division of labor. So what is that division of labor means? Some kind of cells perform one function, another group of cells perform one function, another group of cells performs similar different functions. But remember, they will not form any tissue. They will only perform the cellular functions only. They will not perform any different kind of tissues. That's why this porifera are coming under in cellular level of organization. Understand what is cellular level of organization? Only cells are going to present. Cells are only performs the body all functions, right? So the next category is tissue level. As we know, there are the thing is group of cells together to form a tissue. So if the animals is performing is based on tissues, so that level of organism is called as tissue level. So you can see cell performs similar functions to form a tissue. This is the advanced character. This is the advanced character of animals. So that was this tissue level. After that, organ level. So a group of tissues together to form a particular organ to perform a simple similar function. So that forms organ system level of classification. So you can see in the tissue level, the best example is coelentrates, and organ system level platyhelminthes, right? And next one is organ level is platyhelminthes, organ system level is from annelida to chordata. From annelida to chordata, everyone is having organ system. Different types of systems are going to form, but here only cells are going to form, and next tissues are formed, and next organs are formed. So this is how the organism is evolved, right? If you see that starting there is one cell, right? After that only all organisms are going to arrive from a single cell. So that's how starting cellular level is came. After that some modifications happen, the organism is evolved into tissue level. After that the organism is evolved into organ level. After that the organism is evolved into organ system level. So we studied 4 different types of level of organization: cellular, tissue, organ system. And you have to remember the each terminologies. You see, some shows division of labor. They will ask which level of organization forms cellular division of labor, cellular level of organization, and which level of organization perform tissues, tissue level of organization, or otherwise they will ask examples. Porifera performs what level of organization? Cellular level of organization. Platyhelminthes perform what level of organization? Organ level of organization. And next, from Annelida or Chordata or Echinodermata, what performs the organ system level of organization? So you have to remember examples. The main important thing is examples and what category it is. Understand? So this is about level of organization. In this level of organization, as we studied organ system, right? So there are systems, what the digestive system, circulatory system, different types of systems are going to form. But in digestive system and circulatory system also there are 2 types. So digestive system is classified based on the type of thing, is classified into incomplete digestive system, complete digestive system. So based on digestive system also we classified the animals into 2 categories. One is incomplete, those animals having incomplete digestive system, and those animals having complete digestive system. So what is incomplete, what is complete, we will see. So for example, if you take one animal, if the animal is having one opening, if the animal is having one opening, single opening, and this from this single opening only it going to perform the functions of mouth and anus. So that animals, that digestive system is called as incomplete digestive system. We are complete digestive system animals. So in our structure we have 2 different types of openings. One is for mouth, another one is for anus. So those animals having single opening for mouth and anus, for mouth and anus they have only single opening. So those animals having incomplete digestive system, right? So those animals having complete digestive system means 2 openings. Remember, 2 openings, mouth and anus are 2 different openings are there. So those animals are called as having complete digestive system. So see example Ascalemanthus to Chordatas, and you see example Platyhelminthes. So why I mentioned Platyhelminthes here? Anyone can you tell why I mentioned only Platyhelminthes, not no, not Porifera, not Coelenterata. Why only mention Platyhelminthes? Right. So in as we studied in level of organization, organs are going to form in first in Platyhelminthes, not Porifera, not Coelenterata. That's why I kept digestive system. That too is incomplete. It is not completely formed. Incomplete starting individuals. So that's why Platyhelminth is the example for incomplete digestive system. After Platyhelminthes, the digestive system was completely developed and coming to complete digestive system. Likewise, circulatory system is also there. Based on circulatory system also, we classified the animals into 2 categories. One is open type, one is closed type. So what is open type and closed type? For example, if you see, for example, this is, let's take is an example of heart. So from the heart there are arteries and veins. So these arteries and veins are opened into the body sac. Right? This is the body. This is heart. So these are the arteries and veins. So these arteries or veins are opened into body sac only. This is the cavity of the body. If the arteries or veins opens into the cavity, that type of system is called as open type of the circulatory system. For example, if it is not open, this is heart, right? This is tissue. It is not opened into body sac. It directly connected to tissue, and there exchange of O2 and CO2 takes place. Again the blood is going to heart. So deoxygenated blood coming to the, deoxygenated blood coming to the heart and oxygenated blood coming to the tissue. It is like closed. So the blood is not opened into the body sac. The blood is not opened into the cavity. So it is closed. It is encoded by capillaries, where I mentioned, see, capillaries. See, the important point is the difference between open type and closed type. Here there is no capillaries, only arteries, veins are present. But here the capillaries are there. Because of the capillaries only, the blood is directly entering into tissue. Right? Understand? The blood is entering into the tissue. So that's why it is called as closed type. So it is highly regulated process. In body sac, the blood is coming into body sac and the blood is going to mix, and again the arteries takes the blood. So but in closed type, it's a very highly accurate. You can see the blood performs particular O2 and CO2 transportation, right? So this is two types. One is open type and closed type. In open type, blood fills in tissue spaces. Closed type, blood circulates through vessels, arteries, veins and capillaries. The main difference is here there is no capillaries, here capillaries are present. So this is the level of organization. So based on digestive system also, circulatory system also, we classified animals into different categories. Digestive system we classified complete digestive system, incomplete digestive system. Circulatory system, open type and closed type. So you have to remember what is open type, what is closed type, what is incomplete digestive system, complete digestive system. So when we discussed in non-chordates, chordates, I will tell directly porifera is incomplete. Porifera is an open type of circulatory system, or coelenterata is open type of circulatory system. I will tell directly only that statement. Then you have to remember what is open type, what is closed type, what is incomplete, what is complete. So that's why I am telling here only. So that's why we are studying basics of classification. When you understand the basics of classification, then you will understand easily chordates and non-chordates, right? So this is level 1, levels of organization. So next one is about symmetry. Next fundamental feature is about symmetry. So what is symmetry? So when you take an animal and when you cut an animal, if it is forming two equal parts, then the animal is on symmetry. If it is not having two equal parts, then is asymmetry. Here I mentioned, see, asymmetry, A means no. So there is no plane divides the body into two equal halves. For example, you see sponges. Sponges are the example for the asymmetry, right? So if you see, if you cut the sponges into two parts, it is not going to form two equal parts. Anyway, the sponges does not form any two equal parts. So that's why sponges are in asymmetry. Coming to the symmetry, as I said, if the animal is going to form two equal parts, that is symmetry. In symmetry also there are two types. One is radial symmetry and bilateral symmetry. In radial symmetry, what happens now? The animal is going to form two equal parts. There are many planes. So you can see there are any plane. There are so many planes. For example, this is the example of Hydra. Hydra, you can see this is one plane, this is another plane, this is third plane. There are three different planes. These all three planes are going to cut the animal Hydra into two equal parts. So more than one plane is going to cut the animal into two different parts means it is radial symmetry. If only one plane is going to form the animal into two different equal parts means it is bilateral symmetry. Remember, radial symmetry nothing but many number of planes. The example is coelentrates, ctenophora. It is very important. Not echinodermata, adult echinoderms. Remember, adult echinoderms. Then what about larval echinoderms? Larval echinoderms are bilateral symmetry. This is the NCERT direct question. Can you switch on the AC? AC. Right? So that was the most important thing. Adult echinoderms are radial symmetry, whereas larval echinoderms are bilateral symmetry. This is the one of the question asked in one NEET examinations, remember. And bilateral symmetry are annelids, arthropods, molluscans, chordates. These are the examples for bilateral symmetry. Only exception is larval form of echinodermata. Understand? So these are the different types of body symmetry. One is asymmetry. Asymmetry example is sponges. Next one is Radial symmetry. Radial symmetry example is adult echinoderms. Remember adult echinoderms. And coming to the bilateral symmetry, Annelida to chordata. In between there is a larval form of echinodermata, right? Remember. And understand what is radial symmetry. Radial symmetry is nothing but any plane can cut the animal into equal parts, two equal parts. Whereas coming to the bilateral symmetry, only one plane is going to cut the animal into two equal parts. Understand? So this is about body of symmetry. This is the, it is classified based on the symmetry. Animals are classified into three different classes. Next one, based on the number of germ layers. You see, based on number of germ layers also we classified the animals into two different categories. One is diploblastic and next one is triploblastic. See what we will see what is germ layers. So germ layers are nothing but, so when fertilization happens, zygote forms, right? After zygote, what happens? Blastocyst is going to form. After blastocyst, gastrula is going to form. So in this gastrula, what happens? In this gastrula, what happens? In gastrula, there are three layers: ectoderm, mesoderm, endoderm. There are three germ layers. We called it as germ layers. So what happens? For what these germ layers are going to present? From these each germ layer, there are different types of tissues are going to form. For example, ectoderm is going to form the skin or nervous system, or mesoderm is going to form the blood or visceral organs. Endoderm is going to form gut digestive system of our body. So that's how each and every layer is classified. Ectoderm is going to form some kind of tissues. Endoderm is going to form some kind of tissues. Endoderm is going to form some kind of tissues, whereas mesoderm is going to form some kind of tissues. So these are the germ layers which are present in embryonic development only. From that germ layers, whole organism is going to form. So based on the presence of number of germ layers, for example, in animals there are only two germ layers. So those animals having only two germ layers are called as diploblastic animals. So what germ layers are they are going to have? Only ectoderm and endoderm. So they are only having, see, they are only having ectoderm and endoderm. This ectoderm gives rise to epidermis, whereas this endoderm gives rise to gastrodermis, lining of the gut, right? So the animals having two germ layers, so those animals are called as diploblastic animals. Can I say the animals having three germ layers, they are called as triploblastic? Right? See, the animals having three germ layers, ectoderm, mesoderm, endoderm. So those animals are called as triploblastic animals. You can see ectoderm is going to form skin, hair, neurons, nails and teeth, whereas mesoderm is going to form muscles, bones and heart. So generally you will get heart is from which layer the heart is developed? Mesoderm. Acid mesodermal origin. You will get some kind of question also, no problem. Next one is endoderm. Endoderm is gut, lungs, liver. So from endoderms, these organs are going to form. From mesoderm, these organs are going to form. Ectoderm, skin and neurons are going to form. So the animals having three germ layers are called as triploblastic. The animals having two germ layers are called as diploblastic. Right? You can, this is the picture. You can see this is ectoderm. This is actually this is diploblastic, right? There is no mesoderm, and instead of mesoderm there is a tissue called mesoglia is present, that not a tissue. Ectoderm, endoderm is going to present, whereas in triploblastic animals ectoderm is there, mesoderm is there, endoderm is there. Understand? So that was the NCERT picture in that is present in your textbook, right? So this is about number of germ layers. So what fundamental feature? Which one? Third one, right? First one we already discussed, level of organization. Second one, body symmetry. Third one, number of germ layers. Understand up to clear? Right? Oh. So the next one is nature of coelom. Next fundamental feature is nature of coelom. So we have to know what is coelom. Anyone know what is coelom? Yeah, body cavity. So body cavity which is present inside our body. For example, let's say where it is present. So this is the body, this is the alimentary canal. In between alimentary canal and outer layer, the cavity, all the thing is called as body cavity. So the body cavity is filled with an coelom, which is filled with something jelly-like substance that called as coelom. So based on the coelom also, we classify the animals into different categories. If the body is having cavity, coelomates. If the body does not having any cavity, we call it acoelomates. Simple, A means no, there is no coelom, so that's why they are called as acoelomates. So there are animals, there are acoelomates. Example, flatworms, also called as flatworms, right? So they don't have any cavity, right? So if they are having cavity, which is called as coelomates, then coelomates also classified into two different classes. One is pseudocoelomates, another one is eucoelomates. Pseudo, I will tell, false. If you see here, there is no cavity. Endoderm is there, mesoderm, ectoderm is there, mesoderm is there, and this is endoderm is there. There is no cavity. This is the example for acoelomates. Whereas if you see this thing, ectoderm is there, endoderm is there, mesoderm is also there, but the cavity is not lined by the mesoderm. So how it has to be there means, see, this is ectoderm, this is mesoderm, this is endoderm. This space is cavity. See, upper side means ectoderm side also mesoderm is there, endoderm side also mesoderm is there. The cavity is fully lined by mesodermal tissue. You see, the cavity is fully lined by mesodermal tissue. Then this type of animals we called it as eucoelomates. If it is fully lined by mesoderm, it is fully lined by mesoderm means eucoelomates. Eu means true. Eu means true. If the animal is if the animal cavity is fully lined by mesoderm means eucoelomates. But here if you see, the cavity is not fully lined by mesoderm, right? So it was formed like pouches, scattered pouches. So that it is having cavity but not fully lined by mesoderm. So that's why it's called as pseudocoelomates. See, the body cavity not fully lined by the mesoderm. That was an important point. If the body cavity is not fully lined by the mesoderm means those are pseudocoelomates. If the body cavity is completely lined by the mesoderm means eucoelomates. If the animal does not having any cavity means acoelomates. Understand? In eucoelomates also there are two categories. One is schizocoelomate and enterocoelomate. So how the name schizocoelomate, enterocoelomate given means based on their formation, how they formed the coelomate. Based on the formation they gave. Schizocoelomate, this coelom is formed by the splitting of the mesoderm. Understand? For example, you see here the mesodermal tissue at the starting origin only they split it into two sides, right? For example, here the mesoderm is arised from archenteron. Archenteron is nothing but gut. Archenteron is going to form alimentary canal. Right? If the mesoderm is split into two equal and that is going to form coelom means schizocoelomates. If the mesoderm is arised from the pouches of archenteron, that coelomates are called as enterocoelomates. Remember these terminologies. Schizocoelomates is nothing but their mesoderm is split into two equal and from that only the animals is going to form, coelom is going to form. Whereas in enterocoelomates, mesodermal pouches from the mesodermal pouches only coelom is going to form. Then those animals are having enterocoelomates. Understand? So this is about the nature of coelom. If the animal is having coelom, coelomates. If the animal is not having body cavity, acoelomates. If the animal is having coelom means they are classified into two different classes. One is pseudocoelom and eucoelom. Yet pseudocoelom means pouches are there, eucoelom means completely lined by mesoderm. In eucoelomates also there are two classes, one is schizocoelomates and enterocoelomates. Schizocoelomates are formed by splitting of the mesoderm, enterocoelomates formed by pouches of the mesoderm, right? Understand, pouches of the archenteron. Archenteron is nothing but the gut, alimentary canal. In embryo we called it as archenteron. In future the archenteron is going to form digestive system. Understand? So this is about nature of coelom. Understand? Right. And the next one is body segmentation. So generally the body is divided into different categories, right? Head, tail, trunk, like that. So in smaller organisms also the body is lined or divided into small segments. It is a serial segment, serial repeating units. So those repeating units are called as segments. Understand? Those repeating units are called as segments. So based on these segments also we classify the animals into two different classes. One is segmented animals, unsegmented animals. If you see Ascaris rumblicoids, it is long structure. There is no segments are going to present in Ascaris lumbricoids. But for example, if you see some nereis or earthworms, so they are having some segments, right? So based on the segment also we classified the animals into unsegmented animals, segmented animals. So in segmented also there are two categories. One is metameric segmentation and normal segmentation. So these two animals, these two segmentations are going to seen in only few animals. For example, metameric segmentation is the one of the prominent feature for annelids. So what is metameric segmentation? So what metameric segmentation means, the inside internally divided and externally divided. So the body is divided externally and internally. For example, see, this is the metameric segmentation of annelid. So externally we can see the cut. Internally also when you cut the annelid, you will see internal cut also there, internal segment is also there. But coming to this arthropoda, only you can see external segments. Internally they are all fine. Internally the whole cavity is normal cavity, normal chamber. But in annelids, internally also you will see the segment pattern is there. So that's how metameric segmentation. Metameric segmentation is nothing but externally, internally segments are going to visible. So that type of segmentation is metameric segmentation. This is only present in annelida. Remember, metameric segmentation only present in annelids. Whereas in arthropods, segmentation is only there. Normal segmentation is only. Before that, all are unsegmented animals. After that also mollusk is also unsegmented animal, right? Echinodermata is also unsegmented animal. So only segmentation we can see in annelids, arthropods. Annelids having metameric segmentation, arthropods having normal segmentation. So that is about body segmentation, right? Understand? Shall I move? Right. Next one is body plan. In this body plan, you will see there are three different cell aggregate plan, blind sac, tube within tube. So what is body plan? Nothing but how the body is made up of, how it's look like, simply how it's look like. So the first simple form is cell aggregate form. We already discussed this cell aggregation in which animal? Porifera. Porifera is made up of only cells, right? So the cells only is going to form animal sponges. So that only perform all different kind of functions. So the body sac, only cell aggregates, only cells are going to present, means that plant is called a cell aggregate plant, which can seen in sponges. And example of sponges, there are cycon and spongilla. And the second type of plant is blind sac plant, which is only present in cnidarians and platyhelminthes. There is nothing tube, there is nothing digestive systems, there is nothing so-called complex organism. There is only one animal, inside there is a small cavity is there, sac is there. So that kind of organization is called as blind sac, that only present in coelenterate and platyhelminthes. After that what happens? After that organ system of level classification came. Organs are going to form, digestive system is going to form. So that's how when digestive system is there, there is a tube. Digestive system is nothing but tube. So the animal is The animal is one tube. Inside digestive system is also one tube. So that type of animal plan is called as tube within the tube, right? From nematodes to everything from chordata up to chordata. So that type of plan is called as tube within tube plan. So this is normal simple the body inside cavity, that's only. So that is blind sac plan. There is no cavity, only cells are present means cell aggregate plan. Understand? This is normal terminology, it is not present in NCERT, but you have to remember they ask this question in your examination, so that's why I mentioned. So you can see this is the example for cell aggregate plan, only cells are present, whereas this is blind sac, there is body, only sac is there, only cavity is there. So this is simply blind sac plan. But here if you see the body is kind of one tube inside the endoderm, so this thing digestive system is also like one tube. So that's why these animals are called as tube within tube plan. Understand, there are three, one is cell aggregate, blind sac and tube within tube. Next, notochord. Notochord. So what is notochord? It is a structural support of an animal. Without notochord, the animal is not going to present in a some kind of structure, right? So you can see it is a mesodermal origin. As I mentioned, there are three germ layers. Mesoderm is going to form this notochord structure. It gives the structural support to the animal during development. So in our we don't have notochord. The notochord is replaced by vertebral column, the spiny thing. So likewise, based on the presence of notochord, absence of notochord, we classify the animals into two different categories. One is non-chordates and chordates. So non-chordates having no notochord, whereas chordates having notochord. See, the animals without chordata, without notochord, those are called as non-chordates. Examples are poriferans to hemichordata. Whereas the animals having notochord, those are called as chordates. So they are from cephalochordates to mammals. Up to us we have chordates, but in our chordates this notochord is replaced by vertebral column. Understand? So based on the presence of notochord, we classify the animals into non-chordates and chordates. Right. So this is the classification, this is the summary. You see, cellular level is going to present in porifera, and tissue organ system are going to present from coelenterata to chordata. In that coelenterata, ctenophora consists tissue level of organization, platyhelminthes organ, and here organ system level of organization. Whereas the without body cavity acoelomates are platyhelminthes. Platyhelminthes does not have any coelom. After that, pseudocoelom is ascalmenthes. True coelom is starting from annelida. Remember, from annelida, true coelom. True coelom is nothing but eucoelomates. Eucoelomates is starting from annelida. Likewise, symmetry. Symmetry is example for coelenterata and ctenophora, and also adult echinoderms. Bilateral symmetry from platyhelminthes chordata in echinodermata larval form of echinodermata. So this is the final summary of the basics of classification. So now you understand what is symmetry, right? What is segmentation, what is level of organization, what is diploblastic, triploblastic, incomplete and complete, open and closed type of circulation, coelom, and next what? Chordata and non-chordates body plan. So with all these, if you study the non-chordates means it will be very simple. You will understand very clearly. I hope all of you understand all these terminologies, right? Right? So with this, we will continue to non-chordates. Right? Before going to non-chordates, I want to mention, so what are the non-chordates? Phylums are there. So these are the different phylums that are going to present in non-chordates up to from porifera, starting with porifera to chordata. So these are phylums. Remember, these are all phylums. In chordata, we have different classes, aves, reptiles, all are classes. Some people will think a mammal is a phylum. Okay, aves is phylum. Generally most of the people remember like that only, but chordata is phylum, all are classes. Remember phylum Porifera, Coelenterata, Ctenophora, Platyhelminthes, Aschelminthes, Annelida, Arthropoda, Mollusca, Echinodermata, Hemichordata, Chordata. So we are going to study each and every phylum with an example. So I am telling that how to remember different characteristics of a particular phylum. So the thing is, you have to at least remember one example. For example, phylum Porifera means sponges, Coelenterata means hydra, and Ctenophora means pleurobranchia, and Platyhelminthes earthworm, Aschelminthes nematodes, Annelida, Arthropoda, there are a lot of insects are there, Mollusca, phyla, shells, and octopus also you can remember, no problem, and Echinodermata, starfish, Hemichordata, Balanoglossus. Chordata we will discuss. So if you, when you remember the characters with an example, you will definitely never forget. So whenever you see the sponges, you will definitely remember all the characteristics of phylum Porifera. Whenever you see hydra, you will definitely recognize all the characteristics of Coelenterata. So my suggestion is that, so when you, if you want to remember the characteristic of phylum, just remember one example of the particular phylum, and based on that example, based on the diagram of the sponges, just write all the characteristics of the sponges what are the present in phylum. So that's how you can remember in all the characters of non-chordates. So in this session we are going to discuss up to Hemichordata. After that, next class we discuss chordata, right? Let's start. Phylum 1, Porifera. Right. So as I said, phylum Porifera, the best example is sponges. With this diagram, if you remember all the characters, it's very simple, right? So in every slide I mentioned the phylum names and also the name of the phylum, how it is derived, why it is called as phylum Porifera, like that you just mentioned. The phylum Porifera is nothing but porous means pore, and the fera means beer. So these are pore bearing animals. How they are called pore bearing animals means, for example, if you see the example of one sponge, this is the body of sponge. In this body of sponge you can see these are the different pores are present. So that's why this phylum animals are called as porifera animals. So what are these pores? These are the entry sites which are called as ostia. Remember, these are called as, scientifically these are called as ostia. From this ostia only water is going to enter into the body, right, into the body of the sponge. And how they are going to exit? Exit is coming to osculum. Understand, this is entry, this structure is called as osculum, these pores are called as ostia, right? And as I said, this sponges is asymmetry. As you know, whenever you cut the sponge, you definitely know there is no two equal parts, asymmetry, and they are cellular level of organization. And also is the thing is, they will show division of labor or not, right? Cellular level means they will show division of labor. Cellular level of organization, asymmetry. They are generally marine. There are also freshwater sponges like spongula is there, spongula is freshwater sponge. So generally marine habitat, and also freshwater sponges are also present, right? So as I said, ostia and osculum. So what purpose this ostia and osculum? This is for water feeding. Water is enter into the body of the sponges through ostia, and water is exit to outside to osculum. So this system is called as canal system. Remember, this is the one of the top important point, canal system. Another system is there that is coming under in Echinodermata, where you get another type of system. In sponges you will have canal system. In canal system, what are there? Ostia and osculum. Ostia is responsible for entry into the water, whereas osculum is responsible for exits. So what purpose this canal system is there? This is for food gathering, respiration and excretion also. Canal system is responsible for this. And coming to that, this sponges, in this body of the sponge there are some cells. These cells are called as choanocytes, also called as collar cells. For what these choanocytes or collar cells are present means, they helps in water entry, water currents. They helps the water has to enter into the body. For that purpose these choanocytes are present. You see, flagellated cells lining the spongiocele canals helps in water current and food capture. They will capture the food, they will allow the water inside the body. For that purpose only choanocytes are present. So you see, this is outer layer. Inside this all cavity is called as spongiocele. The body of the cavity of the sponges is called as spongiocele. To entry, for this the water has to enter into the spongiocele, right? So for that purpose only these choanocytes, so called collar cells, are going to present in this sponges. Understand, the sponges important point is they are having choanocytes, they are having canal system, they are asymmetry animals, they are cellular level animals. They are, it is also one of the important point, they are generally marine, they are not mostly. Sometimes they will ask they are mostly marine. No, they are not mostly marine, they are generally marine, because there are some freshwater sponges also present. So they are generally marine, freshwater sponges also there. You have to remember what is the example for the freshwater sponge, right? So the next one is, so that digestion is intracellular. So what is intracellular and extracellular digestion? Intracellular digestion is nothing but the food is going to enter into the cell. Understand, the food is going to enter into the cell. For example, take this is a cell having nucleus and all the digestive enzymes, right? The food is going to enter into the cell means it is called as intracellular digestion. Whereas if the digestive systems are releasing out to the cell and the digestion happens outside of the cell means extracellular digestion. In these animals the digestion is intracellular. In higher level of animals the digestion is extracellular digestion. We have extracellular digestion because we secrete enzymes, right? We secrete enzymes into the stomach. There the digestion is going to happen. So that's why in our we have extracellular digestion. Inside these small animals they are having intracellular digestion. Intracellular is nothing but the cells are having digestive systems are not releasing any enzymes into the body cavity. The food is going to enter into the cell, the digestion happens inside the cell. Whereas the extracellular digestion means digestion happens outside the cell. So that was the difference between intracellular digestion, extracellular digestion. Sponges are having intracellular digestion. The next one is endoskeleton is made up of, supported by spicules and spongyl fibers. This is also important statement. It is made up of spicules and spongyl fibers. So next one is reproduction. Coming to reproduction, so these are hermaphrodite. Hermaphrodite is nothing but both male and female are going to present in one single organism. So those animals are called as hermaphrodite. Internal fertilization, inside the body only the fertilization is going to happen. So that was called as internal fertilization. Whereas external fertilization means the fertilization happens outside the body. Understand, if the fertilization fusion of male gamete and female gamete is happens outside the body means extracellular fertilization. If the fertilization happens inside the body means internal fertilization. And the development is indirect. Indirect means it will not directly give an individual like resembles like adult. There are some stages called larval stages. Larval stages are going to present in this animal. So you have to remember what are the larval stages: parenchyma, amphiblastula. So these are the two larval stages that are going to present in this porifera. So the important thing is we have to remember all the larval stages of all phylums. For example, in porifera means parenchyma and amphiblastula, and next phylum there are different types of larval stages. You have to remember each and every larval forms of all phylum animals, right? it also reproduce asexually with the help of fragmentation. Fragmentation, you know, right? Chopping, right? With the help of fragmentation also, it will also reproduce asexually. Sexually, it will produce, there are hermaphrodite is there, internal fertilization is there, indirect development is there. You all know, right? Internal fertilization, indirect development. Understand internal fertilization, indirect development. So then the final one is examples. You see, spongilla is the freshwater sponge. Exception, generally marine, but freshwater sponges are also present. So what is the freshwater spongilla? They generally they'll ask scientific names and common names. They will not only ask from porifera. They will mention porifera, coelenterata, all these things, echinodermata. They will combine everything and they will mention spongilla and freshwater sponge in another column, match the followings. So you have to remember all the example scientific names also, common name also. Understand? So this is all about porifera. In porifera, what we read? They are generally marine, freshwater spongilla, and also coming to this symmetry, we have to remember symmetry also, most important, symmetry and organ level, cellular level. And what are the important points? Important points here is nothing but canal system. Canal system is only present in this porifera only. There is no canal system is going to present in not every phylum. Only canal system is only porifera. And choanocytes also special characteristic feature is only present in this porifera. Next one, this spicules and spongyl fibers are also present in only this porifera, not everything. And coming to this sexual reproduction, sexual and asexual, in this we have to remember larval stages, parenchymal and amphiblastula. I will tell, I am telling that most of the important points only, which is coming to your examination. And last one is examples. So these are the points you have to remember for your examination. Except this, they won't ask any question. Understand? So this is about phylum porifera. Okay, understand? We'll move into coelenterata. The next phylum is coelenterata. Coelenterata, why these animals are called as Cnidaria? So why these animals are all called as Cnidaria? Because they have some different structures which are called as cnidocytes, cnidoblast. See, this is the one of the key feature. There you have choanocytes. In porifera you have choanocytes. In coelenterata you have cnidoblast, cnidocytes, which contains nematocytes. So this is the example of cnidoblast. This is nematocyte. This is the structure of cnidoblast. This is the structure of cnidoblast, and this cnidoblast contains nematocytes. This is the key structure that was only present in this coelenterata. So that's why this coelenterata is also called as Cnidaria. Understand? So for example, if you see, this is the example of coelenterata hydra. There are so many tentacles are there, right? On these tentacles, these cnidocytes are going to present. Understand? On this tentacle, for example, you see, on these tentacles, these cnidocytes are going to present. It look like needle-like cells. So that's why coelenterata, needle stinging cells. These are called as needle stinging cells. Also called as Cnidaria. Why they are called as Cnidaria means they are having cnidocytes, cnidoblast, and these cnidoplates having nematocyte. So that's why these animals are called as Cnidaria. And where these cnidocytes are going to present means on the tentacles. For what purpose these cnidoblasts, cnidocytes are going to present means, here I mentioned they are anchorage, defense, prey capture. Their anchor is nothing but holding. They are for holding something, and also defense and prey, attacking for the animals, food capture. For this thing, these cnidoblasts are going to present. And also you have to study this thing. Phylum coelenterata having radial symmetry, as I mentioned, organization, tissue level, and thing is diploblastic. You have to remember what is diploblastic. Ectoderm, endoderm, tissue level, radial symmetry, any plane it will cut. If you see any plane, if it is cylindrical shape, any plane it is going to cut into two equal lobes. So you have to remember those points. They are mostly marine. Poriferans are generally marine, coelentrators are mostly marine. So these are the examples. Physalia, Portuguese man of war, Adamisia, sea anemone, Penantula, sea pen, Medrian, brain coral, Gorgonia, sea fan. All the examples are very important. All the common names are also very important. You have to study, you have to remember, definitely you have to remember. Right? This is the one of the prominent feature, what that is nidocytes. They will ask. Understand? So the next. Anyway, it is going to present in your textbook also. This PPT is whole about your NCERT textbook. From NCERT textbook only I made this PPT for explaining yourself. And coming to the body structure of coelenterata, coelenterata is having gastrovascular cavity. This is also one of the key feature where the gastrovascular cavity is going to present. Coelenterata only. Coelenterata only having gastrovascular cavity. And coming to, it is having single opening. Single opening is nothing but incomplete digestion, right? It is having single opening cavity, it is incomplete digestion. Coming to the digestion, it is performing extracellular and intracellular. I also said what is extracellular, what is intracellular. And also the cavity, so the shell of the coat of the coelenterata is made up of calcium carbonate. The skeleton, the bony structure is made up of calcium carbonate. Is also one of the important point. It's a separate note you have to mention. So the body cavity is having gastrovascular cavity, and also it is incomplete digestion, which is having only single opening, and those opening is called as hypostome or mouth. Mouth or hypostome, both are same. And coming to the digestion, it perform both extracellular and intracellular digestion, and the bone is made up of calcium carbonate. Understand? Whereas coming to the sponges, I was to mention point. In sponges also, this endoskeleton spicules are also having two categories. One is siliceous spicules and calcareous spicules. Means some spicules are made up of calcium carbonate, some spicules are made up of silicon dioxide. That was the point I missed, right? So spicules are having two types in sponges, made up of calcium carbonate, also made up of silicon dioxide also. Whereas in coelenterata, it is exclusively made up of calcium carbonate. Understand? I also mentioned the examples, Physalia, all these things. And next one, this is the one of the important feature that is present in coelenterata. What is that? Polymorphism. Polymorphism, metagenesis. There are two kinds of coelenterata. One is polyp and medusae. So polyps are nothing but, you see, this is the example of polyp. Polyps are like stagnant, attached to the substratum, whereas some animals are free living. These are free living animals, they are having umbrella shape. Umbrella shape like animals are called as medusa, they are free living, whereas these stagnant animals are called as polyp. So there are two kind of animals, you see. Polyp, they are sessile. Sessile means stagnant to one pet, and they are cylindrical. Examples of polyps are Hydra and Adamisia. That's also important. And coming to the medusae, medusa are umbrella like shape, they are free swimming. You can see they are not attached to any substratum, they are like normally free moving. So the examples of medusa are Aurelia and jellyfishes. So there is one of the feature which is called as alternation of generation, which is called as metagenesis. What happens in this metagenesis means this medusa sexually reproduce to polyp, sexually reproduce to form a polyp, whereas this polyp, this is sexually. whereas this polyp is asexually. So one animal, for example, let's take Obelia. Obelia is the one of the animal, it having two different forms, polyp also, medusa also. So whenever it is in form of medusa, it is formed sexually to polyp. Whenever it is in polyp, it is formed medusa through asexually. So this is called as alternation of generation. The word given for alternation of generation is metagenesis. Polyp forms asexually, medusa forms sexually. This umbrella structure is called as medusa. This medusa is going to form polyp through sexually, whereas this polyp is going to form medusa through asexually. This remember, they will ask opposite. Medusa form polyp asexually, polyp form medusa asexually. This is wrong statement. Just remember, medusa is going to form polyp through sexually, polyp is going to form medusa through asexually. This alternation of generation is called as metagenesis. Metagenesis is only seen in Coelenterata. Or this polymorphism, this having two different types of body forms, this polymorphism is only seen in Coelenterata. These are the two important points that are going to present only in Coelenterata only. Understand? In Coelenterata what we discussed, metagenesis and polymorphism, gastrovascular cavity and also nidoblast. These are the key features of the Coelenterata. They will ask mostly this question only. And examples. Examples you have to remember. In every phylum, examples you have to remember, and characteristic features, prominent features you have to remember, and this thing common, very common, you have to remember symmetry, organization. When you remember phylum, Porifera means symmetry, Coelenterata, radial symmetry, like that you have to remember everything, symmetry and organization. If there are any characteristic features are there, you have to remember the characteristic features and also examples. Other than this, there is no required. Right? We completed two phylums. One is Porifera, another one is Coelenterata. Now, Ctenophora. Cteno means comb, phora means bearing. Comb bearing animals. These Ctenophora are called as comb bearing animals. For example, if you see this animal, this is Pleurobranchia. This is the one of the important example in this Coelenterata. Every time they will ask the example of Coelenterata is Pleurobranchia. They will mention only this Pleurobranchia and they will ask what this phylum belongs to, what this animal belongs to which phylum, or they will ask characteristics of Pleurobranchia. They will ask what are the different forms, that if they are having different forms, what are the different forms have there in Pleurobranchia. So remember, Pleurobranchia is the only one example in your NCERT that is under Ctenophora. So if you see this Pleurobranchia, here you see these are different types of plates. How many? 1, 2, 3, 4, 5, 6, 7, 8. 8 plates are going to present in this Pleurobranchia. These plates are called as comb plates, ciliated comb plates. One of the important point, characteristic feature only seen in this Coelenterata. So you can see here on these comb plates, cilia are going to like present. So this is one of the important characteristic feature. 8 comb ciliated plates where these kind of feature is going to present means Coelenterata. Right? Understand? Sir, what purpose this thing? This is for locomotion. Why? Because cilia is for what? Locomotion. So these 8 comb plates are going for locomotion only. They will ask, these 8 ciliated comb plates are feeding, digestion. They will mention different examples like options, but the actual answer is for locomotion. This is the one of the feature. Another prominent feature is bioluminescence. This is one of the characteristic. No phylum in this non-chordata showing bioluminescences except this Coelenterata. So how, sir, how this animal is going to form this bioluminescences character? there are some cells which are called as photocytes. These photocytes are present beneath these comb plates. Beneath these comb plates, the cells are called as photocytes. From the with the help of the photocytes, the animal is going to illuminate some light. So that characteristic feature is called as bioluminescence. Bioluminescence nothing but animals is going to illuminate some light. It's giving some light. So that characteristic feature is bioluminescence, which is well marked in ctenophora only. Understand? That is also second interesting point. And you can see there is no nematocytes. Nematocytes where we read nidaria, nidoblast, nidaria, but here there is no nematocytes. And next one, they have coloblasts, which are called as lasso cells. Porifera have choanocytes, coelenterata have nidoblast, nidocytes, nematocytes. Ctenophora have coloblasts, which are also called as lasso cells for food capture. How to remember? We discussed 3 phylums. In each and every phylum, different cells are going to present. You have to remember those cells only, right? Here also digestion is extracellular, intracellular. It's not very important, but you have to remember digestion is extracellular, intracellular. I am mentioning this locomotion, 8 ciliated comb plates, very important. Bioluminescence, very important. Coloblast also very important, right? Coming to the general features, these are exclusively marine, right? Ctenophora and coelenterata are radial symmetry, we already discussed, and it is also tissue level. Mesoglea contains means it is diploblastic animal. It is diploblastic animal, and they are also having amoebocytes. Nothing but these are the cells which are helpful in digestion, excretion, circulation, not circulation, digestion and excretion. For that purpose, these amoebocytes are present. These are undifferentiated tissues. You just remember there are some cells which are performing body physiological functions. So digestive system, excretion, they are helping in that system, right? Right. Exclusively marine we discussed, radial symmetry also we discussed, tissue level, diploblastic animals, and locomotion is by comb plates, bioluminescences, coloblasts. So these are the features of this phylum Ctenophora, right? Another thing, coming to the reproduction. Reproduction is by hermaphrodite, only sexual reproduction is there, whereas in Porifera sexual is there, asexual is there. Whereas it is external fertilization, remember it is external fertilization, and also indirect development. Indirect development means there are some larval stages. So the example of larva is sidipid larva, right? In Porifera we have amphiblastula and parenchyma larva. In this Ctenophora we have sidipid larva. So the example of Ctenophora is plurobranchia. This is the only one example which is given in your NCRT. Only one example, that is plurobranchia, also called as sea gooseberry. Understand? Sidipid larva. Done na? I forgot to mention this point. Planula larva in coelenterata. Porifera we are having amphiblastula, parenchyma. Ctenophora having planula larva. Coelenterata having planula larva. Ctenophora having sidipid larva. Every phylum having different larval forms. We have to remember different larval names and also examples and also characteristic features, right? So this is coelenterata planula, Ctenophora sidipid larva, right? So the larval name, hermaphrodite, means two sexes are going to present in one animal, right? If the animal contains male and female gonads means hermaphrodite. If the animal is having only one sex means unisexual, different, right? Ctenophora complete. Any doubts in Ctenophora or the coelenterata or Porifera? Everything is clear, right? I hope. Porifera, Coelenterata, Ctenophora. Right. So now we are going to start with next phylum, Platyhelminthes. Platyhelminthes. The name is derived. The name is simply platy means flat. Helminthes means worms. So these are called as flatworms. Simply you can remember platyhelminthes means flatworms. Why they are called as flatworms? Because the body is dorso-ventrally flattened. Body is dorso-ventrally flattened. So what is dorsal, what is ventral? So this is the part of dorsal and the back side is ventral. So likewise the animal is also, the bottom side is ventral, the upper side is dorsal. Understand? So the dorsal and ventral is normal flat like structure. So that's why these animals called as flatworms. So because of the presence of this dorso-ventrally flattened structure, this platyhelminth is called as flatworms. Understand? And the thing is, this is a bilateral symmetry. See, I was mentioned first triploblastic. Up to we discussed only diploblastic animals. So the platyhelminths, the key feature is these are the first triploblastic animals. And also, we, as you know, these are acoelomates. And organ level is also started from this platyhelminths. So we have to remember this. And here you see this is the one of the key feature. These platyhelminths you can see hooks and suckers. Hooks and suckers are going to present in which phylum? Platyhelminths. These are for attachment to the host tissues because these are parasites. So parasites are going to present on the surface of the animal. They have to attach the tissue. They have to attach the tissue, and they have to suck the blood from the parasites, so-called host. So that's why these hooks and suckers are going to present in this platyhelminths, right? So what happens? They absorb nutrients directly to the body surfaces with the help of these hooks and suckers, right? So the digestive system is incomplete, which means only one single opening. The excretion and osmoregulation by flame cells. This is the one of the important point. Excretion and osmoregulation is done by flame cells. Excretion, you know, osmoregulation is nothing but what? The maintenance of sodium. The maintenance of sodium and water balance in the body is called as osmoregulation. That was maintained by these flame cells, which are also called as protonephridia. Remember, flame cells. Protonephridia structure is going to present in this platyhelminths only. Flame cells, one of the key cells which are present in only platyhelminths, responsible for excretion and osmoregulation. Another one of the important point is the nervous system is ladder-like. Ladder-like nervous system is going to present in which phylum? They will ask this question. That answer is platyhelminths. These are very important points. Next one is reproduction. Coming to reproduction, there are hermaphrodites. Fertilization is internal fertilization. Inside the body, the fertilization will happen. Development is indirect, which means larval stages is there. What are the larval stages? They have so many larval stages: miracidium, sporocyst, rhedia, cercaria. So these are the example of different larval names of the platyhelminths. Remember, platyhelminths have many number of larval stages. Examples: miracidium, sporocyst, rhedia, and cercaria, and also metacercaria also there. So these are the examples of the larval forms of platyhelminths. Next one, it is the one of the key feature, high regeneration capacity. For example, let's take this planaria. This is example of planaria. If you cut this planaria, next day you come, you see two planarias are there. From this part, the upper part is going to form. From this part, the lower part is going to form. So that's why it has high regeneration capacity. Regeneration capacity means the lost tissue, it is going to form with itself only. But we don't have any regeneration capacity. Only this thing. Lower organisms. Lower organisms, everyone is having regeneration capacity. In that, high regeneration capacity is only seen in platyhelminthes, that too exceptional planaria. Remember? So these are the key features: high regeneration capacity, ladder-like cells, flame cells, hooks and suckers, first triploblastic, bilateral. So all these are the key features of platyhelminthes. Understand? This thing, special adaptation like parasites or acoelomates, dorsoventrally flattened, these are like normal statements you can remember, but the most important thing is first triploblastic animals, and hooks and suckers are going to present, and coming to this flame cells and ladder like nervous system and high regeneration capacity. These are the prominent key features that are present in this phylum Platyhelminthes. And you see the examples. There are 3 examples in your NCERT: Taenia solium, Fasciola hepatica, Schizostoma. Scientific names and common names. You have to remember the scientific names and common names also, right? You have completed 4 phylums: Porifera, Coelenterata, Cnidaria and Platyhelminthes. Understand Platyhelminthes? Is there any doubt? Tapeworm, liver fluke, planaria. So this is the upper view. When you see the side view, the dorsal and below ventral are dorsoventrally flattened. So that's how you remember, okay, these platyhelminthes are dorsoventrally flattened. So that's why these are called as flatworms, and they are the first triploblastic animals. You have to remember each and every point by seeing the animal image. You see, there is one thing, these are also segments, right? But I explained only metameric segment and normal segments, but you see these are not true segments, not true segments. So that's why these are called as what? Not true means pseudo segmentation. Pseudo segmentation is present in which phylum? Platyhelminthes. That is also one of the important point you just mentioned. Pseudo segmentation is prominently found in Platyhelminthes only. Metameric segmentation prominently found in Annelida. Normal segmentation formed in Arthropoda. Remember? Right. So the next one, Aschelminthes. Aschelminthes is also called as Aschelminthes is also called as Nematihelminthes or nematodes. They are aquatic and terrestrial. Habitat is nothing but where it is going to present, the environment of that animal. And the body is cylindrical, round. See, this is the most important thing. When you take cross section, it is like round. When you take cross section, if you see, it is like round. So that's why these are called as roundworms. They mention in one question they mention Aschelminthes are called as roundworms. Statement 1. In statement 2 they mention they have tube within the tube. Statement 2. Tube within tube. Okay, but the thing is, because of the presence of tube within the tube, they are not called as roundworms. Why they are called as roundworms? Because when you cut the animal cross section, you see the round kind of appearance. So that's why they are called as roundworms. Both statements are right, but statement 2 is not correct explanation of statement A. Right? So why they are called as roundworm? Because of the cross section. Understand? So the bilateral symmetry, triploblastic. Important points to pseudocoelomate, and the one important point is first organ system level. We already discussed first organ system level. First organ is Platyhelminthes. First organ system level is Aschelminthes. First triploblastic is Platyhelminthes. Here also triploblastic is there. From here triploblastic is going to continue. Pseudocoelom. Pseudocoelom is only present in this Aschelminthes. Next true coelom will come. Right? So these are also very important. See, this is unsegmented. Platyhelminthes is what? Pseudo segment. Whereas this is unsegment. There is a transparent protective cuticle called collagenous. So on this surface there is a collagenous layer is going to present on the body of the nematodes. Coming to this, the alimentary canal is complete, which means mouth and anus is going to present. And next one is excretion is performed through renate glands, whereas in platforms what cells? Flame cells. You have to remember, platforms having flame cells. Excretion is performed with the help of flame cells. Osmoregulation is done with the flame cells, whereas in nematodes, renate glands. Sexes are separate, dioecious. You mentioned up to here we studied hermaphrodite. Both animals, both male and female are going to present in one individual. From here, sexes are separate, right? Sexual dimorphism, which means by looking you can identify what is male, what is female. You see, long one is female, short and curved is male. By looking you can easily identify what is male and what is female. So that term is called as sexual dimorphism. Larger female, smaller males. Understand? Next one is internal fertilization, mostly oviparous. Example is Ascaris. Some are oviviviparous. Example is Urchularia. So do you know what is oviparous, oviviviparous? Right? Egg laying individuals are called as oviparous. Their direct developing is called as viviparous. But some animals that are egg laying, but they are present inside the body cavity. Understand? They are laying eggs, but the eggs are present inside the body cavity of the mother. So those animals are called as oviviviparous. Example, Urchularia branchofti. Understand? Right. And direct or indirect development, if it is indirect development, there should be larval form. So the larval form is rhabditiform larva. Each and every phylum there are larval forms are there. Larval forms are very important. Right. So these are the examples for Ascalemanthus nematodes. So what are the examples? Ascaris lumbricoides, Enterobulus vermiculus, Urchularia branchofti, Ancylostoma duodenale. These are the common names. You have to remember scientific names and common names also. Right. We discussed Ascalemanthus. The important points are cross-sectional, roundworms, first organ system, unsegmented. You have to remember all these things. Renate glands. In Platyhelminthes, flame cells. So when you compare with the other phylums, then you will remember all the different characteristics, right? So that's why I am explaining previous things also. Renate glands, their flame cells. Next, sexual dimorphism up to Platyhelminthes we discussed hermaphrodite. But here sexual dimorphism is there, and mostly oviparous, some are oviviviparous. The important thing is rhabditiform larva. Larval stage is rhabditiform larva. Examples. So this is all about Ascalemanthus. We discussed Porifera, Coelenterata, Ctenophora, Platyhelminthes, Ascalemanthus. 5 phylums we discussed, right? Right. We discuss another phylum and then we will do some PYQs of this thing. So the thing is, next one is annelids. Right, 6th one. Annelids. Annelids are called, annulus are called as ring leda form. These are for ringworms. These are called ringworms. They have ring-like structures on their body. So that's why they are called as ringworms. Understand? They are aquatic, terrestrial, free living. These are the habitats of that phylum. And this thing, bilateral, triploblastic is also we know. These are the first true coelomes. Ascalemanthus pseudocoelom, Platyhelminthes acoelomates. The first true coelom is going to form from here, annelids. So that's why this is most important point, first true coelom. In true coelom we have two different types of coelomates. One is schizocoelomate, another one is enterocoelomate. In that first one is schizocoelomates. It is having schizocoelomate. Next one, the body organization is organ system level of organization. In Aschelminthis only we have first organ system. From Aschelminthis all the animals are organ system level of organization only. Organ system level, first true coelom, triploblastic, bilateral. This is the common features of this annelids. And coming to this prominent features, what is that? Metamerism, which is called as metameric segmentation. I already mentioned externally, internally the body is divided, segmented. So that thing is called as metameric segmentation. That phenomenon is called as metamerism. This is the one of the important feature is going to present in this annelida. Understand? Body is elongated, divided into segments externally and internally. As you know, internal segmentation divided by septa. Septa is nothing but a layer. Right. And the thing is locomotion. Locomotion is done with the help of different structures. Here I mentioned different structures and what are the structures present in different animals. For example, parapodia, it is present in this Nereis, whereas setae present in earthworms, suckers are present in leeches. So they are the structures which are helpful in locomotion present in different animals. You have to remember different structural names and the structure where it is present, Nereis, earthworm and leech, right. Next one, circulation is closed type. Nervous system, paired ganglia and double ventral nerve cord. This is the most important point. I will explain. We have, as I said, we have notochords, right. Notochords, as you know, it is mesodermal origin. It is a long tube. It is forming the body structural back support. Then what is nerve cord? Nerve cord is nothing but it is a rod-like structure which is going to build up with neurons. Neurons. It is made up of neurons. It is rod-like structure. It is arised from ectoderm. Remember, it is originated from ectoderm, whereas notochord is originated from mesoderm. Remember, and as I said, for example, this is an animal. This is ventral. This is dorsal. So, right. So this non-chordates are having nerve cord on ventral side. Nerve cord on ventral side. Whereas this chordates, our humans, we are having nervous system on dorsal side, and notochord is also present on dorsal side. So that was the difference between notochords and non-chordates and chordates. Non-chordates having this nervous system on what side means ventral side, whereas in chordates nervous system is going to present on what side means dorsal side. So that's why this double ventral nerve cord is present in annelida. Understand? Here the excretion in osmoregulation is performed with the help of nephridia. In Platyhelminthes, flame cells. Understand? Aschelminthis, rennet glands. And then coming to the reproduction, sexual reproduction is there. Monoecious, which means earthworm, dioecious, Nereis and leech. Fertilization is external, internal fertilization also there. Development is direct is there, indirect is there. If it is indirect, larval form is trochophore larva. Both types are there. Direct development is there, indirect development is there, external fertilization is there, internal fertilization is there. And coming to monoecious, unisexual is there, bisexual is there, hermaphrodite is there, earthworm is hermaphrodite, and separate sexual dimorphism is seen in Nereis and leech. You have to remember this example, trochophore larva. Examples of annelida, Ferithema, Nereis and Hirudinaria. The scientific names of that examples and common names of examples. Understand? So this is all about annelids. Understand? Annelid, let me revise annelid important points. First, true coelom, cygocoelomate. This metamerism, metameric segmentation, this phenomenon is called as metamerism. And locomotion structures, parapodia, setae and suckers. Paired ganglia, double ventral nerve cord. We have only single dorsal nerve cord. We have, chordates have single dorsal nerve cord, whereas this non-cord is double ventral nerve cord. And excretion osmoregulation is nephridia, and reproduction is normal. Sexual reproduction is there only, monoecious, dioecious, external, internal, direct, indirect. If it is indirect, definitely the larval form is trochophore larva. And finally examples. So this is all about annelida. Annelida are called as ringworms, right? So platyhelminthes flat forms, ascalmenthes roundworms, these are ringworms. So this is all about annelida. So let's see few questions of your NEET and EAPCET. Those are very simple questions only, which is yeah, you can see anyone can do 29. Not read, understand the question. Planaria possesses high degree of regeneration capacity. Yes? Yeah, planaria is having high regeneration capacity, whereas coming to this reason are planaria exhibit polyembryony. As I mentioned, anything polyembryony, right? So the answer is C. Yes is correct, R is not correct. Good. The next one, 35. Coelomates. This is the level of organization questions. All right, this basic classification questions. First, we'll go with platyhelminthes is what? Platyhelminthes is acoelomates. Next, mollusca, chordata, enterocoelomates. So D is what? 1. Yeah, D is 1. D is 1. Option you were telling. Okay. A is 3. Nematoda is pseudocoelomate. Mollusca is cytocoelomate. And chordata is, sorry, platyhelminth is acoelomate. The answer is B. So just they simply ask phylum and what are the coelom. Next time they will ask phylum, what are the level of organization. Phylum, what are the thing of embryonic development layers. Phylums, examples. Phylums, some excretion cells. For example, they mentioned platyhelminthes, they mentioned flame cells. Mollusca, rasping organ. Nematoda, rennetic glands. They will mention likewise. So, okay, this is B and that is C. Understand the question, right? So, this how statements related questions come, and simple matching questions also will come. So, very simple scoring unit it is. Next one, wait. Okay, this platyhelminthes we already discussed. We will discuss later. First, see this 25th question. We already discussed this platyhelminthes, right? So these are first bilateral symmetry triploblastic animals. Next, they have pseudocoelom with acts as like hydroskeleton. Platyhelminth is what? So you can definitely remove this question. Identify the correct statements. So if you remove fertilization is internal, development is direct with very nervous. This statement is correct to every phylum, I think, right? So fertilization is generally internal or indirect. There in every larval, in every phylums, larval forms are there. So you can hold it. So if you eliminate B from 3 options, the remaining option is A, C, D. So they are first bilaterally symmetrical and first triploblastic animals also. And hooks and suckers, as you know, hooks and suckers are present. A, C, D. So this is from platyhelminthes question. So that's how next time they will ask nematyhelminthes, and next time nematodes. Likewise, they mention some statements. They give like the statements. They ask the answer. Oh, sorry. Ctenophora, Nematoda, Platyhelminthes we discussed. We didn't discuss this thing, Echinodermata. But you can answer. It's very simple question. A is lazo cells and bioluminescence. Bioluminescence is exhibited only in Ctenophora animals, right? D4, right? Right. Echinodermata, adult echinodermitas are radial symmetry, but as larvals are bilateral symmetry. So based on this, what is the option? D4. C4, right? A2 ra. B5 Renetic glands with amphids. C1 Platyhelminthes acoelomates. D4. So the option is C. So here they mentioned renetic glands with radial symmetry. Nematodes does not have any radial symmetry, right? So renetic glands with aphids. Radial symmetry is only seen in what? Coelenterata, Ctenophora, Echinodermata. Radial symmetry is seen in Coelenterata, Ctenophora, Echinodermata. This nematoda does not have any radial symmetry. It is bilateral symmetry. So that's how this is one mistake. It is not 3, it is 5. Okay, C option. ఇది కూడా చేస్తారా? Obelia, Cipher, Adamasia, Cyanimo, Penantula, Cpen. With this option you can directly choose C. Penantula is Cpen, right? So this is also very important simple question. So whenever you see matching question means it's a very simple question. You have to do perfectly. See, we are in competitive, right? So if you are in competitive field means most of the paper, if 50% of the paper is coming simple means you have to attempt all the simple questions. If the question is tough means definitely the topper will also definitely confuse, the topper will also make mistakes, but the topper will not make mistake on simple questions. So if you don't make any mistakes in simple questions, then you will be in competitive. If you are making mistakes in simple questions, then you will be eliminated from the field. So don't give mistakes in simple questions. If it is very tough kind of question means then you can eliminate that question, you can leave the question. If it is very simple, you have to definitely answer with accurately, perfectly, right? So these matching questions are very simple. Next one. Oh, sorry. That is arthropoda. You can oh, you are mentioning. Not present. I am asking not present. Where this metameric segmentation is seen? Annelida. So this is not present in arthropoda. As I already mentioned, right, there are locomotor structures are present, parapodia, setae, right? Jointed appendix, arthropoda, Patinus exoskeleton. This thing you can, in case of porifera, the spongiocele is lined with flagellated cells. Choanocytes. Choanocytes are the present like on the body wall. So they are helpful in entry of the water, they are filter feedings. Ostia are the pores which are the openings of the body. Osculum is the exit side. Choanocytes are the thing which are present on lined with the flagellated cells. So that was the answer for this 139 question. This is notochord. This is also another question. You can answer this. 174. So they asked choose the correct answers. This is not correct. Poriferans are not pseudocoelomates. Ascalementhes are acoelomates. Ascalementhes are pseudocoelomates. Platyhelminthes are acoelomates. Simple question. Just you have to remember the statements only, right? So that's how you can answer 168 also. Just now I know told where right. So this radial symmetry is found in Ctenophora, Coelenterata, Echinodermata, not present in this hemichordata. That is NEET question, NEET 2023. Just a simple question they asked, NEET level. That we'll discuss. You can answer, but you have to analyze the question first of all. For this question, you have to analyze the question. Then you can only make the answer. And that's also very simple question. While trying to find out the character of a newly found animal, they discovered one animal. They are studying that animal. A researcher did the histology of adult animal and observed a cavity. There is a cavity with the presence of mesodermal tissue towards the body wall, but no mesodermal tissue was observed towards the alimentary canal. So what it be? Right. So he observed a cavity, means it's not a acoelomate, that it will be definitely coelomate. There is no spongocoelomate like kind of structure. Schizocoelomate is nothing but a true coelo which is having splitting. Here they mention they observed a cavity with the presence of mesodermal tissue, but no mesodermal tissue was observed towards alimentary canal. So that's it's like there is a cavity, but mesoderm tissue is present towards the body wall, but not alimentary canal. So that is like pouch kind of structure. So that is pseudocoelomate. So that's how this is 2025 question. They ask some little tricky kind of thing, but it is a very simple question. So this is how the different questions are coming in your NEET and EAPCET examination. We discussed only up to what? Annelida. We discussed only up to Annelida. We completed Porifera, Coelenterata, Ctenophora, Platyhelminthes, Nematohelminthes and Annelida. So from Arthropoda we will discuss tomorrow class Arthropoda to Hemichordata, and we will discuss some PYQs, and next class we discuss Chordata. I hope all of you understand up to Annelida. If there is any doubt, you can comment, you can ask me if there are any doubts in up to Annelida. Is there anything? Is there nothing? Okay. If there any doubt, you can ask, no problem. Just I will revise just one thing. In Porifera canal system, choanocytes, this larval forms are there, spicules. In Coelenterata, nidocytes, examples, Coelenterata, gastrovascular cavity, and this alternation of generations. This is also very important in Coelenterata. Ctenophora, bioluminescence, 8 ciliated comb plates, lazo cells, coloblast. For what? Food capture. They will ask lazoblasts for locomotion. No, lazoblasts for food capture. Hey, please. The larval form of this Ctenophora. Platyhelminthes, hooks and suckers, first triploblastic animal. The next one is high regeneration capacity planaria, flame cells, ladder-like nervous system. Ascaleminth is roundworm because of the cross section. First organ system level unsegmented. Rhabditiform larva, sexual dimorphism, renate glands. So these are the key points you have to remember. Annelida, schizocoeloid, first true coelom, and metamerism, metameric segmentation present in Annelida only, right? Parapodia cetus occurs are present. Double ventral nerve cord, paired ganglia, and trochophore larva. So these are the points you have to remember for your examination. Okay, we will continue the next class tomorrow from this arthropoda. Right? Understand? Okay, done. Jointed appendages.